So a little bit different entry quiz this time. I was asking you which concepts you want to learn most about. I'm hoping that we're going to move in a particular direction to make sure that we're answering the questions in class that most of you want to hear the answers to. So what I'd love to pilot, we're going to try this. Before each class, if you have questions based on the videos watched before class and so forth, email them to me. And then I can incorporate those so that we can ask everybody in class of the 3 or 4 top questions that I received from you, which ones do most of you want to hear the answers to? Make it a little bit more of a democratic process. And I did keep saying that I was going to talk about these various things in class. You saw yesterday a Blackboard announcement with details about how to do all of these processes. So I'm going to leave you to read those details and ask questions about them as you need. And the only thing I need to point out really on this slide is that for the Comedy, Genius, and Current Genetics, you only need to do one of those, 1x. So you get one presentation of either type can earn you up to the full 7 points. So you don't have to do both. You don't have to do either, of course. But.
For the 7 points, if let's just say we got a 5 out of 7 on the current genetics,
yep.
Can we make those other 2 points up with comedy?
Yeah.
Okay.
If that were the case.
Well,
yep.
If anyone needs help finding comedy stuff, there's a Facebook group called Trust Me, I'm a Biologist, and it's amazing. They have a lot of great stuff on there.
Yeah. But remember, the hope is that you come up with something, not that you share something that somebody else has created.
Still good.
Yes, still good entertainment. Other questions about this stuff? Okay. Sweet. So then we get to the pre-lecture video. The one question that I asked you to think about was, how do you make a cross that produces individuals that only have homozygous genotypes? Does anybody have a solution to this question?
All A1s or all A2s? Like A1, 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 A1?
Okay, so here, homogeneous, A1, homozygous. So when you say all A1, let's talk diploid. So we've got A1 over A1. There's a homozygous diploid. We've got the same version of gene A. They're homozygous for it. What would be the goal then? You need to find another genotype to cross. What would be the genotype of an F1 that would be the solution to this problem, given what we've got right now?
Also an A1, A1?
So an F1 generation has to be homozygous. So we've started with one parent that has A1s. So the F1 has to, all of the F1, not just one F1, every single F1 organism has to have that genotype. Which parent did that version of the chromosome come from? Pick one.
Mom.
You say mother? Yeah. Okay, so let's say, which version of allele A1, the one on the left or the one on the right, came from mom into her offspring?
The one on the right?
Okay, the one on the right. Does it matter?
No.
They're both the same, so it doesn't matter. We're just guessing. No one up here actually knows. We're just picking randomly. Okay. So if this is the case, if that individual got that version of the chromosome from their mother, then what does the genotype of the father have to look like?
The same.
Okay, so it's got to have at least one A1 because
F1 is homozygous.
there had to be an A1 to contribute to the offspring to produce that genotype. So now what do we know about the other chromosome in a diploid? What could we put there? A1? We could. What about A2, a second allele of the gene A? Could dad be A1 over A2?
But not all.
No, because then you wouldn't have, sorry.
No, that's fine.
Then you wouldn't have only
Based on the Punnett square, it should come out right.
Right. So the question was, what's the key word in the question that tells us what to put in here? Only. The word only. So what genotype would we have to have the dad to be to ensure that any gamete produced by that mom and that dad produce homozygous offspring? Has to be A1. If it was A2, what would occasionally happen in the F1 generation?
There would be heterozygous.
Dad would contribute that allele to his offspring. Mom would definitely give what? A1. So occasionally you'd get an A1 over A2 offspring. That's not homozygous. Therefore, we know that that's not the genotype that dad had. So that's actually pretty boring. It's an A1 over A1 by A1 over A1 cross. So the take-home message, it's really hard, or really easy, depending on your perspective, to make an entire generation of individuals that have the exact same genotype. You have to start with two parents that are homozygous for a single allele. Yeah.
So, I mean, this is kind of the approach in dog breeding, right? If you wanted to get certain traits or maybe create a pug, you would have.
What are you having against pug? Anyway, sorry.
No, I love my own one. She's just a genetic disaster.
Oh dear. That's harsh, but fair, I guess.
Yeah, so essentially, like you're looking for these traits, and the more homozygous, the easier it is to select for these.
To
traits.
keep those traits going. So this is often what geneticists that are doing conventional breeding want to do. You want to find crosses and produce organisms that are. If you want to generate a trait and keep it around, you want heterozygotes or homozygotes? Homozygotes for this reason, because then you can breed them together and you know that there's no genetic variation. When there's only A1s in the population or the organisms you're working with, then you know for sure that their offspring are only going to be homozygous for that allele and have that trait. Fortunately, we humans know that inbreeding is not a good idea, right? But we still do it with dog breeding and plants and all sorts of other pigeons. Any pigeon fanciers in the audience? Mice people? Rat people? No? Yeah? Okay. We'll look at pigeon genetics someday, maybe. So more application of our knowledge. Based on the question that we asked last class, making a green fluorescent rolling worm, sell them at raves, we'll put them on as bracelets. What genotypes what are all of the genotypes, let's list them all, that would produce that sort of a worm? How many different genotypes could produce a worm with these two traits, phenotypes? Right, we've got genotypes and we've got phenotypes. Genotypes is the description of the genes, phenotypes is what the organism looks like. So what's one way to make a green fluorescent rolling worm? So we've got, let's do the nomenclature first. What nomenclature did we use for GFP? That was an asterisk, I think. So that was an allele that produces GFP. The other option was plus, which is wild type. And then there were minus and plus alleles for roller. So what two genotypes can make you green? Here on the left side. Asterisk over plus, because you only need one version of the GFP gene, asterisk, to make you green. So what's the other option? Homozygous. Got two copies of the GFP gene, one on each of your two chromosomes. Diploid individual. Okay, so that's what's happening at one locus, wherever the GFP gene is located. Either homozygous or you're heterozygous. You've got at least one copy of GFP. What about the roller locus? Does anybody remember what genotype you have to be a roller? It's got to be homozygous. So how many different genotypes can create this phenotype in the bottom left that we're looking for? 2. How did you get 2? We've written 3 genotypes up here. So we're looking at combinations of the genotypes. So you either get the top genotype or the bottom genotype over there. Either one of those has to be combined with this thing on the right side to make it a rolling worm. So if you're green, you have either genotype 1 or 2 on the left. If you're a roller, you have to have the genotype over here on the right. So how would we write that? It'd be asterisk over plus, minus over minus, or asterisk over asterisk, minus over minus. Then the question is, what punctuation do I put in between them? Well, we don't know if they're on the same chromosome or not. A semicolon would tell us they were on different chromosomes. We don't know if they're on different chromosomes or not. I haven't told you that. So in class, for us, we're going to just use a single dot in between the 2 genotypes, which tells us the only information that gives us is we don't know where in the genome these genes are located. Same chromosome or 2 different chromosomes. We don't know. Which one of those 2 genotypes would be better if you want to make sure that all of the worms and all the effort you're going through to make all these green fluorescent rolling worms, make sure all of their offspring are the same phenotype. They're all green and they all spin around when they crawl. You want the top genotype or the bottom genotype? So why the bottom? Right, it's homozygous genotype. There's no way, there are no pluses, there are no wild type versions of the gene in this genotype down here. Right? There's 2 asterisks and 2 minuses. If you took that hermaphrodite, remember these worms have self-fertile hermaphrodite sexes, if you took one worm and you selfed it, and it was GFP over GFP minus over minus, or asterisk over asterisk minus over minus, the only types of offspring she could produce by herself would be identical genotypes. She's clonally reproduced she. I don't know if that's an appropriate term for hermaphrodite, but when I say she, imagine I'm saying S/he. Right? So the benefit of homozygosity, what geneticists that are doing traditional breeding often look for, creating a homozygous genotype that will produce only individuals that have the same traits or the same phenotypes in subsequent generations.
Dr. Ross?
Yeah.
So if the green glowing worm has an asterisk and only one minus, would it still roll?
No.
No.
You have to have 2, by definition, have to have 2 minuses at the roller gene. One on a mutant version on each of the 2 chromosomes to exhibit that trait, that phenotype.
Dr. Ross?
Yeah.
So the answer to what are all of the possible genotypes that would produce a green roller, do you only want us to write the homozygous ones? The asteroid and asteroid?
No, in that case, both. So 2.
So it would be asteroid, asteroid, and negative, negative.
Let's just focus on the bottom question here. Given these 2 starting genotypes, what's the most efficient way to get to green rolling worms? So what I'd like you to do is to talk to a neighbor the next 3 minutes or so. We've just talked about how to combine these 2 traits into an organism. We're going to do it by crossing. You've got a strain that's got roller genotypes, you've got a strain that's green fluorescent. How can you make those 2 individuals together to combine both of the traits? It's not going to be sufficient to get to the first generation. What's going to happen if we cross these individuals?
Are you saying that the rollers are are not GFP and the GFP are not rollers?
That is what you should assume based on the way I wrote these genotypes. It's a fantastic question. So think about it this way. For each of these individuals, we're talking about two different genes. We're talking about the GFP gene, and we're talking about the gene that causes rolling. So even though when I've only tell you, and anytime, this is always going to be true, if I give you a genotype, if I haven't given you information, you should assume that an individual is homozygous for the wild type versions of the gene. So in that case, what's the genotype of the roller worm at the GFP locus? It's plus over plus. So that rolling worm has no GFP genes, and likewise, the GFP worm is plus over plus. So those are the complete genotypes of the worms that we're dealing with. So take a couple minutes, see if you can figure out how to cross them together. What's the first step?
Produce an F1 generation.
Okay, produce an F1 generation. So you self which side?
The right side.
Or do you cross these two together? You cross or you self? Not do you self, but do we self these worms? Well, what happens if you self the worm on the right? Do you make any worms that are rollers? Will you ever make any worms that are rollers? Because there's no minus allele in that individual. That doesn't mean this is the wrong approach or the approach, not the approach that I'm looking for, but that's a problem. The worm on the right by itself will never produce a roller worm. Will the worm on the left ever produce a green worm by itself? No. So we have to combine these two sets of genes at some point. So let's do it in the first generation. Uh-oh. Now we've got to talk about Punnett squares and what these mean and what they represent. You already watched a video on this, supposedly. So what goes across the top axis? The top of the box. What are we going to write up there? Something about genes, something about one of the parents. Excellent. So let's put this parent's something about their genes across the top. Let's focus on GFP first. This will take us through into next class, by the way, which is not Monday. Don't come here on Monday. I will not be here. You can come if you want. The door will be closed and I will not be here. Maybe you prefer that. I don't know. So let's focus on GFP. What's written across the top is genotypes or haplotypes. We want to know gametes. We want to know what are the genotypes of the gametes that this parent produces. Those genotypes of gametes is the definition of a haplotype. So for GFP, what are the two different versions of gametes that this parent on the right will produce?
Asteroid and plus.
Asterisk and plus. Meiosis, using this genotype, half of the gametes will have asterisks and half will have pluses. And as I explained, that's why we divide that line exactly in half. And divide this Punnett square exactly in half, because exactly half of this parent's gametes are going to have one of the versions of the chromosome, and the other half of the gametes are going to have the other one. I'm going to call this parent B. So the parent B gametes, that's what you write on the top and down the side of the Punnett square.
Wait, didn't you just say that was
Haplotypes. Yep. The gametes, the genotypes of their gametes, the haplotypes. And we're just looking at the GFP gene right now. Next class, we're going to talk about adding in the roller locus. So what about the other parent? It's GFP. It's only got one allele. They're both chromosomes, 2 chromosomes, diploid individual, 1 chromosome's wild type, the other chromosome's wild type. So half of their sperm or eggs have plus, and the other half have plus. Because there's no asterisk over here. Right. So what should I do to this Punnett square?
Not divide it, just put the plus. You don't need to divide it into.
So you could divide it into,
But it would give you the same amount, right?
but there's only one type of gamete that's being produced for that gene by that parent, parent A. All of the gonads, or all of the gametes, its gonad produces only gametes that contain plus. So the reason we divide Punnett squares in half is because of what aspect of biology about these imaginary organisms we're talking about. Why is it divided in half vertically and in half horizontally?
Equal percentages of each,
Heterozygosity, equal percentages,
Ratio?
ratio.
it's the ratio, half. So why is it half? Why is it half one gamete and half the other? What essential concept in biology or in genetics tells us?
They got half of their genetics from their parents.
You only have 2 with a dominant.
We're talking about diploids. 2 copies of each chromosome. So there are 2 possible, maximum of 2 possible haplotypes in an organism. Right? That's why most Punnett squares, when you're talking about one gene, in a diploid, you divide it in half vertically and in half horizontally. Because there's only 2 possible haplotypes that can come out of either parent. Maximum 2, minimum 1. As in the case on the left here, GFP plus over plus, I guarantee it, you know that all of their gametes have a plus allele of that gene. Hence the point that we could not bother with that horizontal line. Because 100% of the gametes have plus. So that makes a 2-cell Punnett square. So once we write in the gametes from the parents across the top and down one side, what do? This is the part you remember from any time you've ever heard about this in the past. You fill down, into each cell you fill down the gametes from the top, and you fill across the haplotype of the gamete that's on the left. And that gives you the predicted genotypes of the offspring. And the areas of those 2 squares tell you the frequencies, or how likely you are to see both. So half of this table is full of heterozygotes. So about 50% of the offspring produced by this cross should be heterozygous. And the other half should be homozygous for the wild type. "10 seconds. Talk to your neighbor. What's the genotype ratio resulting from this cross?" Go now. Quick. Genotype ratio. Okay. That was 10 seconds. Time's up. And what's the answer? 2 to 2, 1 to 1, but either same, 2 to 2, 1 to 1, 4 to 4. There are an equal number of individuals with the genotype on the left and the genotype on the right, 50% of each. So for every 1 individual that's heterozygous, the left side, you should have another sibling that has the genotype on the right. And that's just because fertilization happens randomly, people. You can't control which of your sperms and which of your eggs collide. If half of the sperm have 2 different haplotypes, the chances for every fertilization, there's a 50% chance that one or the other haplotype will contact the egg and produce an offspring. Which I guess raises a really, really, really important point. about Punnett squares because we're talking about the random process of fertilization. 15 seconds. Talk to a neighbor. What is the genotype ratio you get out of this cross if there's only one offspring produced? Go.
51, because it'll either be this one or this one.
Okay, that was 15 seconds. Thank you very much for your participation. What's the answer?
1/2. 1.
The ratio is, you got one offspring. Which one's it going to be?
Either one.
Either one. So the ratio is either, if it's the one on the left, it's 1 to 0. So if you're a human and you only have one kid, and you have these genotypes, I don't know if they're rolling humans, but whatever. If you only had one kid, you would either have one or the other. So it'd either be a 1 to 0 ratio or a 0 to 1 ratio. So what do we have to do what is this Punnett square telling us when we see half one genotype and half the other? It's a probability that you get for every offspring that you look at is the probability. We've got a 50-50 chance of any one offspring being that genotype on the right or that genotype on the left. So to actually arrive at this ratio, a 50-50 ratio of kids with the left genotype and kids with the right genotype, what do we have to do? Biologically speaking. Have a lot of kids. The more kids you have, the more likely you are that half of them, truly half, have the genotype on the left and half on the right. And that's why we use organisms like C. elegans or fruit flies or other organisms that have lots and lots and lots and lots and lots of offspring, because it's only then when we can actually study is genetics working the way we expect. If you have 324 offspring, do you get 100 oh crap, I can't do that math in my head. Do half of them, or approximately half, have the genotype on the left and the other half on the right? Sure. If you're human, it's harder to do this because if you've got 3 kids, you're never going to see, this is the point, this is an expected ratio. This is a prediction, the Punnett square, the genotypes in the middle. This is what you expect to observe. This is not usually ever what you actually measure as a geneticist. This is an ideal situation. If you had an infinite number of kids, then yes, half would have one genotype, half would have the other. In nature, in reality, that doesn't happen. There's always going to be some slight difference. 49% on the left, 51% on the right, or something like that. It's an important point to consider from now, at least until the end of this class, if not until cell biology and then evolution and then other classes. Yeah.
So now just looking at the roller, which has to be minus.
Yep.
Your F1 generation is going to be heterozygous, so it's not going to be a roller. So you would have to cross 2 heterozygotes in the F2 generation.
So we have to get into future generations to actually solve this question. And we're going to get into, yes, absolutely, we're going to get into the solution to this question during our next class, which is when?
Wednesday.
Next Wednesday. Fantastic. When we'll also get into a couple other questions. But for next Wednesday, we've got one more video to watch. This will help us answer this question. We're going to look at the dihybrid cross, looking at how to combine 2 different genes and their traits into an organism, like GFP and the roller. So there are 3 movies. Hold on just a second. One more, maybe more important announcement. 2 of these bottom 2 movies are much shorter, and they will be online within the next 24 hours. Finally, exams. They've been scored. I'm going to put the scores up online sometime this afternoon. You're probably not going to like your score. Your low score for this semester gets thrown out. It's very likely going to be this exam. I'm going to put the key online. It's a movie. It's on YouTube. I'll make it publicly available. What I ask you to do is to self-assess. What you need to do if you want to grow, which is what I cherish in this class, is that I don't care if you start low, as long as you work to improve by the end of the semester. That's what I really care about. So if you get a bad grade, take heart. That doesn't mean that you've failed, that means that you've got growth to do. That's fine. So what I'd like you to ask you to do to start that growth process is to look at the key online, look at your answers to your exam, because you've all got them. I don't have to hand anything back, because you've got the digital versions somewhere on your computers. You decide where you think you lost points. So if you want to know, if you want assessment on your exam, come talk to me. I'm happy to tell you exactly what. Yeah, you missed points on that question and that question and that question. But I want you to try to figure it out, because that's how you're going to improve, so that when you take the final and you are faced with similar questions, you know where you went wrong initially and you figured out how to solve the problem. So look at the key. Next week, if you want to talk, absolutely, we'll do that. Okay?